Ok, cominciamo. Buongiorno a tutti, benvenuti a questo webinar. Sono Alessandro Schienoni, rappresento Garzani Specialities che oggi con Natural Remedies vi ha invitato a questo webinar dedicato alla gestione dello stress e della risposta immunitaria nei suini con soluzioni naturali. Per chi ancora non la conoscesse, spendo solo due parole su Natural Remedies, che è un'azienda di struttura familiare, di terza generazione, indiana, la sede a Bangalore. Ha scelto molti anni fa la strada della valorizzazione delle piante ayurvediche attraverso studi approfonditi dei loro fitocostituenti e degli usi tradizionali e dell'interazione tra la fisiologia degli animali e queste piante. Alcune di queste piante poi sono state scelte e associate in modo sinergico a costituire i prodotti che oggi utilizziamo regolarmente nel settore zootecnico. Sono tutti prodotti interamente naturali, ovviamente, efficaci e sicuri. Hanno un effetto costante grazie all'esclusiva tecnica di standardizzazione delle piante crude o delle loro parti senza ricorrere all'estrazione. Oggi non possiamo approfondire questo aspetto che costituisce veramente un po' la pietra miliare di questo know-how di Natural Remedies. Parleremo invece di uno di questi prodotti, che è Phytoc o Phytosee, che da diversi anni ci fornisce continue osservazioni e informazioni utili ad allargare poi il campo applicativo verso il benessere animale. Lo farà il dottor Junaid Syed di Natural Remedies, che è in Natural Remedies da 4 anni, veterinario operativo da 8. In Natural Remedies coordina il supporto tecnico e commerciale con alcuni paesi europei, tra cui l'Italia, alcuni paesi dell'area APEC e alcuni paesi dell'Africa. Oggi è con noi per parlare quindi di sostegno immunitario nei suinetti e di come gestire lo stress nei suini in modo naturale. Vedremo ad esempio come con il Phytoc si vengono a modificare le immunoglobuline nel colostro delle scrofe e nel siero dei suinetti, o come per esempio viene controllato il cortisolo nei momenti di maggiore stress. La presentazione durerà circa 45 minuti, durante i quali vi invito a postare domande nella sezione domande e risposte, che vedete a video, fatele in inglese o in italiano a vostra scelta. Al termine della presentazione cercheremo di rispondere, ma per motivi di tempo, se non ce la facciamo, lo faremo in un secondo tempo, ma vi invitiamo a utilizzare anche le mail che vedrete a fine presentazione, così quelle lasciano traccia e poi ci risentiremo più avanti. Quindi adesso vi invito a seguire la presentazione. Junaid, I'm finished. This is your time. I'll let you the stage, please.
Thank you very much, Alessandro, and very good morning to everyone joining us this morning from Italy and from other parts of Europe, and probably from some other parts of the globe like Asia Pacific. So good afternoon to them as well. So on behalf of Natural Remedies and Garzani Specialities Italy, our business partner, I would like to welcome all of you to this webinar on how to boost the piglet immunity and managing the stress naturally in swine. The aim and vision of Natural Remedies and Garzani Specialities Italy is to bring greener solutions to the doorsteps of the Italian consumers. And with this vision, we have joined hands and are in association for more than 10 years now. So without any further ado, I would just like to share my screen with all of you. Alessandro, please let me know if my screen is visible. Okay, thank you. Thanks for the hands up. So as I was saying, that the aim and vision of Natural Remedies and our partner Garzani Specialities is to bring down the greener solutions to the doorsteps of the Italian consumers. And what we are trying to do, we're trying to add a dash of green with profitability for the swine industry of Italy. We believe in a common vision of bringing greener solutions by harnessing nature and applying science to them, so that the quest for the food safety through feed safety is ensured. This is a humble initiative of Natural Remedies to bring forth the solutions which are bringing profitability through the greener solutions. The topic for today's webinar, as you might be knowing, is how to boost the piglet immunity and manage stress in sows naturally. When we talk about stress, or even before beginning to the stress, the quest for the human race has always been to have the healthy food. Healthy human means consuming healthy food. And for healthy food, the most important thing is to have the animals in the best of their health and the best of their ability to perform within the specified limits and standards. When I talk about stress, stress is usually may not be visible to many. And when you talk about stress, there is an old school of thought which is very prominent and says that the most prominent and the most profound form of stress which is seen in animals is the heat stress. It cannot be discounted. Heat stress still forms one of the major forms of stressors to the animals. But with the with more intensive farming, which we are into now, more intensive pig farming, there are other multiple stressors which have come into picture. And the heat stress has become now an old school of thought, and people are now focusing on new forms of stressors. Whatever new forms of stressors are there, all these stressors, what they will do, these stressors will have an impact on the overall natural immunity of the animals, the natural defenses of the animals with these multitude of stressors which these animals are encountering in the intensive farming we are putting them into. It will have an impact on the natural immunity, and thereby it will make them more susceptible to the disease. This vicious cycle will keep on continuing, and what will happen? The result, the outcome would be that the productive performance of these animals will be compromised. The health and well-being of these animals will be compromised. Now, talking about stress, as I mentioned that there are a multitude of stressors other than heat stress or cold stress, or you can say an environmental stress. There are multiple stressors which are present in the current intensive pig farming. If you see on the left-hand side in the red, just give me a moment. If you see the stressors in the red on the left-hand side, these are the stressors which are very commonly observed and seen in the current intensive pig farming. The ones which I have mentioned in the blue are the ones which are very prominent. Now, the most important stressors in pigs, like social stress, is a very important form of stress because pigs have a natural tendency to develop a natural or social hierarchy when they are put in the groups. And when they are put into the groups, the stress level, because of that intent to produce that hierarchy and rank in the herd, leads to the development of stress in these pigs, say it in sows or in piglets, whether in different grouping stages. Similarly, the metabolic stress, which is not so thought of by people, is one of the major forms of stressors. If you see the animals which we are rearing now, the animals, we're forcing them to reach to a particular body weight in a particular specified period of time. The same animal would have taken more time 10 years down the line, and the same animal is now reaching to this particular body weight in a very short time. Now, what has happened? The genetic manipulations and the genetic makeup which we have changed for these animals, we are forcing them to produce a particular body weight in a specified period of time. And what happens? Because of that, we're increasing the metabolism of these animals. And when we increase the metabolism of these animals, the cells become hyperactive. And when cells become hyperactive, there is a metabolic stress which will be incurring on these cells. Formation of more free radicals will be there inside the cells, and they will become old at a very young age. And the ability of these cells to absorb the nourishment will be impaired. Similarly, the third form of stress, which are very important in intensive pig farming, is the environmental stress, which all of us know very prominently. The heat stress, the cold stress, the fluctuations in the environmental temperature leads to a lot of discomfort in the animals, leading to impairment in the productivity and the overall optimal health of these animals. Then there are other forms of stressors like handling stress. We know that when we have to handle these animals prior to slaughter or when we have to transport them into the trucks, a lot of stress is impaired on them, which puts a lot of impact on the carcass value as well. Last but not the least, there are a lot of immunological stresses also, which these animals are prone to. So in short, the point is that there are multiple stressors which these animals are encountering in the intensive pig farming. Now, what is the impact of these stressors? Multiple impacts would be there on the swine. Let's say in terms of performance, yes, the performance would be affected. There will be reduction in the feed intake, probably reduced weight gain. Similarly, there will be an impact on the reproductive abilities of these animals. The immunity of the animal will be compromised. The quality of the meat will also be affected with these multiple stressors coming in the intensive farming and creating a synergistic effect on the ability of these animals to perform. Last but not the least, one very important impact of stress in swine is the impact on the behavior of these animals. So there is increased aggression the moment these animals are grouped together, which leads to problems like tail biting and other vices like ear biting. Now, what is the most important impact of the stress? What is the physiology behind the stress? Whenever the body is encountering these multiple form of stressors, there is a stress response mechanism which is activated inside the body. Two types of stress response mechanisms are activated inside the body. The first and the most important stress response mechanism is the activation of hypothalamus, pituitary, and adrenal axes. And in addition to that, there is an activation of sympathetic nervous system, which also tries to combat this stress which the animal is encountering. Now, what is this HPA axis? Whenever the stress is there, what body tries to do? The body tries to fight against this stress. So there is a hormone which is produced inside the body, which is called as a hormone of fight and flight, like cortisone, corticosterone, and adrenaline. So the hypothalamus is getting activated because of this stress which this animal is undertaking, and it activates the pituitary gland, thereby activating the adrenal, where from these glucocorticoids are secreted. Normally, within a certain limit, these corticosterones or the glucocorticoids are good for the body to reduce the level of stress. But as the amount of stress increases, because the stressors are not dwindling in the farm. In fact, day by day, the amount of stress is increasing. So as the amount of stress increases, this corticosterone level reaches to a certain levels inside the body that it starts producing the deleterious effect on the body by destroying certain vital proteins, antibodies, enzymes, and hormones. In addition to that, there is also one more important axis, which is called as hypothalamus, pituitary, and gonadal axis. Now, this is also one of the responses of body towards stress, that this HPG axis also gets activated, and it has a direct impact on the reproductive ability of the animals. We will see in the coming slides how different form of stressors have an impact on different physiological parameters of the body. Now, let us talk about heat stress. As I mentioned, there are multiple forms of stressors. Let us go one by one into different forms of stressors which are affecting the ability and putting a lot of compromises on the performance of these animals. Let's talk about heat stress. Pigs in general are more susceptible and prone to heat stress. Now, there is a specific reason for that, why pigs are more susceptible, because thermoregulation in animals is majorly done by the transfer of internal heat to the outside. And this is majorly done through panting and sweating through the sweat glands. In case of pigs, the sweat glands are very minimal, and there is a thick layer of subcutaneous fat under the skin, which impairs the loss of this heat through sweating. Now, secondly, the lungs in pigs are comparatively smaller when you compare it with the whole size of the body, thereby making panting also very difficult for pigs to dissipate this excess heat which is produced inside the body. Now, because of this excess heat, there are multiple reactions which will happen inside the body, and although there are multiple parameters which people have to check, like this chart which is here on the downside of this presentation, the Humidex chart, which determines the effective humidity and temperature levels which should be there in the farm. If you see this Humidex chart as well, you will understand and you will see that even at the best possible temperature and humidity combination, still there are certain stressors, or there is an effect of stress because of this heat on these animals. Now, what is the cascade? What happens because of this heat stress? If you go deep into the effects of heat stress, this heat stress gets amplified coupled with the effect of humidity. Whenever there is humidity as well as heat, the stress because of these factors gets amplified. So what will happen when the heat stress will be there? There'll be more hyperventilation. There will be more, carbon dioxide expiration from the body, which will cause an imbalance of, acid-base imbalance. There will be respiratory alkalosis, which in turn will lead to reduction in the feed intake by these animals. And when there is reduction in the feed intake, we can comprehend that there will be deficiency in terms of essential nutrients which are to be required for the overall performance of the animals. So the outcome of this cascade of heat as well as humidity would be the performance will be compromised. There will be poor efficiency, and the profitability of the flock will be of the herd will be compromised. Now, if you see, there are multiple, validations which have been done to see what is the impact of heat stress on the pigs. At different temperatures, you will see that scientists have seen that there is an impact on the average daily gain, average feed intake, even if a little fluctuations in the temperature are there. There is a validation which says that there is a report which says that a 60 kg pig at a temperature of around 25 degrees Celsius already starts to reduce its appetite because of the stress. And when the temperature from 25 to 28 degree is, it is elevated to 28 degrees Celsius, the same pig of 60 kg body weight will have 50% reduction in its appetite. So this is the impact, this heat stress as well as a coupled, this heat stress coupled with humidity will have on the overall performance of the animal. Going beyond, so heat stress, as we mentioned in the beginning as well, heat stress forms one of the major stressors, but it's now an old school of thought. In piglets, or in case of pigs, there are other stressors like social stress. Social stress, when you're mixing these pigs together, as they try to form the social rank and hierarchy in the herd, it will have an impact on the reproductive ability of these animals. If you see in this, in this slide, what we are trying to showcase is that as the amount of psychological and social stress increases, there is an activation of this hypothalamus, pituitary, and adrenal axes. What they will do, there are certain cells in the hypothalamus which are called as KNDY cells, and they secrete certain hormones like neurokinin and dynorphins. Because of these, they inhibit some important reproductive hormones like gonadotropic releasing hormone, which will have an impact on the luteinizing hormone. So luteinizing hormone is important for the animal to increase the levels of estrogen from the ovaries, thereby increasing the ovulation. So when the social stress increases because of the cortisol, there is impact on the gonadotropic releasing hormone, thereby having impact on the luteinizing hormone, and thus it will have an impact on the ovulation of the animals. So the reproductivity or the reproduction capacity of the animals will also be compromised. Similarly, when the amount of stress increases, heat stress, which is coupled with other form of stressors, there is a study which says that these stressors will have an impact on the cell membrane of, the sphingomyelins. Certain cellular structures will be impacted because of this stress. And because of that, some metabolites or compounds will be formed inside the body like this, which is called as ceramide. Now, this ceramide will increase the formation of reactive oxygen species, and it will impact the mitochondrial dysfunctioning. And when there is more mitochondrial dysfunctioning, what will happen? The cycle of apoptosis or programmed cell death will be there. And what will happen at the end, the oocyst functioning will be impaired. So this is again a very important impact, the stress which is coupled with heat, or the heat stress which is coupled with other form of stressors, will have on the functioning or the reproductive functioning of the animals. Going beyond, As I mentioned in the beginning also, one of the most important stressors which impacts the overall, or which is one of the major stressors to these pigs or the current intensive form farming in pigs, is the stress which is created by mixing the animals together. Now we have to mix these animals. Let's say the sows are to be mixed after weaning so that they can be inseminated again. The piglets are weaned and they are grouped together at the time of weaning in the nursery. And we are mixing these sows again for the insemination. Now, during this time, when we are mixing these animals together, there is uh, there is a normal tendency for these animals to showcase uh, a behavior of hierarchy development in the herd. If the sows, for example, which are older in age and are much larger in parity, they are more stronger and try to dominate the sows which are lesser in parity and and younger in age. And because of this, there is an increased aggression which will be observed in these animals, which will lead to more, problems like tail biting, ear biting, bruises, in turn having an impact on the overall reproductive functionality of these animals. Possibly, it will have an impact on the furrowing rate, it will have an impact on the litter size, it will have an impact on the quality of litter. So, group housed sows are more prone to the stress, and this stress has an impact on the overall health and well-being of these animals. Last but not the least, as I mentioned, different impacts of stress in pigs. Very importantly, you know, uh, one of the major impacts of the heat stress or the other form of stresses is on the quality of the meat. When we have to transport these fattening pigs to the slaughterhouse, no matter how sophisticated the transportation system is, there is always an amount of stress which will be implicated on them and which will have an impact on the carcass value of the pigs. Depending on the amount of exposure towards the stress these animals are facing, different types of meat formations will be there. For example, when the animal or these fattening pigs uh, are subjected to the short-term stress. For example, you're handling them very improperly. What will happen? They will lead to more breakdown of glycogen. They will activate the stress mechanism inside the body, wherein the cortisol level will increase, and this cortisol will try to bring more sugar or glucose inside the body. And this glucose is produced by breaking the glycogen inside the muscles as well as in the liver. Now, when this glycogen is broken down to glucose, it finally changes into the lactic acid, and this lactic acid dictates the pH of the muscles inside the body. Because the stress is so high, I mean, this is a short-term stress. The glycogen breakdown is so high, so quick that the lactic acid production is very high. It leads to the formation of certain, uh, you know, uh, quality of meat, which is called as PSE. Certain type of meats, which is called as PSE, pale, soft, and exudative meat. Exudative type of meat, where uh, the meat is more pale, it is less flavored, and it becomes more tough at the time of cooking. Similarly, if the animals are facing the long-term stress, let's say these animals were not slaughtered on the day they were brought to the lairage, they are kept with the other pigs in the lairage. Because of the aggression which they will showcase at the time of grouping, the glycogen will be depleted very quickly. And the moment these pigs are slaughtered the next day, there will be less glycogen to produce lactic acid, and thereby the pH of the muscles will increase, leading to the formation of uh, poor quality meat, which is called as DFD, dry, firm, and dark quality of meat. Both of these are, you know, leading to the reduction in the carcass value and leading to the rejection of the carcass because of the impacts it will have on the quality of the meat. So going to now the piglet immunity, or in fact, before going to the piglet immunity, once we saw all these different kind of stressors, there is a single common link between these multiple stressors, that the moment the animal or the pigs are encountering these stressors, there is a common phenomenon which is taking place inside the body, which is the activation of stress response inside the body by the animals. And one of the important stress responses is the production of cortisol as a response towards the stress. But as I mentioned in the beginning also, as the amount of cortisol keeps on increasing, there is a side, or there is a negative impact which this excess cortisol brings in, and thereby reduces the overall health, well-being, and the productivity of these animals. Coming to the piglet immunity now. So we have seen that the pigs in the gestational stage, in the lactation stage, in the weaning stage, are encountered with multitude of stressors. Now, talking about the piglets and how important is the maintenance of immunity for these piglets. The immune system in piglets starts to develop early on in the gestation, around 30, 42 days of the gestation inside the body. But because the pigs are having a specific type of placenta, which is called as epitheliochorial placenta, the transfer of these immune cells, which are formed inside the sows or inside the pig, is not able to be transferred to the piglets. So the maternally derived antibodies are the only source of immune protection for these piglets at the time of furrowing. There is a literature which says that the passive immunity, which comes from the colostrum as well as from the milk of the sows, reaches to its peak by around 7 days of lactation. And afterwards, till weaning, the acute immunity in the piglets is still not developed. By 28th day, which is like 10 days after weaning, the antibodies which have come from the sows are present inside the body of the piglets. And the acute immunity, the full-blown acute immunity in the piglets is developed at around 42 days, which is like 2 weeks after weaning. So if you see this graph as well, at birth, the passive immunity from the mother to the piglet is the only source of protection to these piglets. And as the time goes on, the maternally derived antibodies starts decreasing. And the acute immunity of the piglets starts by around 2 weeks after weaning. So there is this gap, there is a window of vulnerability for these piglets wherein they are more susceptible to the infections and they are more susceptible to the stress. In addition to that, the maintenance of piglet immunity early on in the life is very essential because of multiple reasons. For example, if the piglet immunity is maintained at a very high level, it means that the mortality in the early phase of the life, from first day to 7th day, when the mortality reaches around 3 to 13% also, it will be minimized. Secondly, what will happen if the piglet immunity is well maintained, the piglets, there will be maximum number of piglets which will reach to the weaning. Last but not the least, if the immunity is maintained during the early life of the piglets, the overall condition and the uniformity of the body weight of these piglets will be there. So it has been said, and it is being said, that good start is always having a good finish. So if you start well with your piglets having good amount of immunity, it will mean that they will be able to reach to the weaning with uniform body condition and body weight, and will pass through this weaning stress very comfortably till they reach to the level of acquiring their immune response through acute immunity, which comes after 2 weeks of weaning, around 42 days. So if we check just give me a moment. So the immunity to the piglets is dependent on the immunity of the sows at the time of gestation or at the time of furrowing. So colostrum and lactogenic maternal immunity is, as I said, is the only source of immunity to the piglets. And this depends on how much better condition the sow is in at the time of furrowing. Since furrowing itself is a very stressful, uh, process for the sows, and as the animal reaches the furrowing date, the amount of stress keeps on increasing. So it is very critical for the farmers to ensure that the sows are in the optimal health and in better nutrition, and the immune levels of the sows are in good state. Thereby, it will ensure that maximum amount of maternally derived antibodies and other immune cells like T cells, B cells, and some hormones or uh, antimicrobial peptides are transferred from colostrum and from milk to the sow, to the piglets. Now, this also depends, as I said, the, the immunity in the piglets depends on the immunity status of the sows prior to the furrowing. And this depends on how better is the nutrition of the sow, how much we are able to maintain and manage the environmental stressors which can impact the well-being of sows at the time of furrowing, and how better nourishment we are giving to the sows at the time of furrowing. So coming to the summary of different stressors which these animals are facing during their lifetime, if you see, there are multiple stressors which have been enlisted here, and how much impact they have on the, let's say, on the parameters like average daily feed intake, daily gain, body weight. And these are not only heat stress related stressors, these are stressors like regrouping stressors, shipping means at the time of transportation, or the dirty environment. All these have an impact on the overall health, well-being of not only the sows, but of the piglets as well. Now, what does body do? The body of the pigs try to maintain a certain form of homeostasis, a physiological equilibrium with its environment. The body tries to resist these multiple stressors. But as we know, there is not a single stressor which is present in the house of the pigs. There are multiple stressors which are present. And because as multiple stressors are present, the ability to resistance to these stressors is compromised and it is reduced, which means the pigs will be more vulnerable to the disease, and there will be more production challenges which will come after the amount of stress increases. Now, this process is called as cross-sensitization. Excuse me. Because of the process of cross-sensitization, where the pigs are not able to resist the multiple stressors which are present, they become more prone and susceptible to the diseases. So what is the requirement? What do these pigs need in order to survive and manage these stressors very efficiently? The point is, we cannot avoid these stressors. These are omnipresent. These are present in the houses. We cannot remove them at all. What we can do is we can manage them with the best possible solutions which are there to manage these stressors more positively. So there is a need to have a solution which will improve upon the endurance of these animals, which will increase the resistance of these animals to resist and fight towards the stress, and thereby will improve upon the natural defense of the animals. They will increase the natural immunity of the animals. Thereby, when both of these will combine, it will impact the overall health and the productive performance of the animals. So with this thought, Natural Remedies came up with this solution of Phytosee, which most of you might have heard of. This Phytosee is called as a natural defense enhancer. It is a standardized botanical powder, which is made up of different types of herbs, from where we are getting a class of phytochemicals which are completely standardized. When you talk about Phytosee and its impact on the animals, we say that Phytosee has 3 different mechanisms of actions. One is the adaptogenic property. The second is the impact on the cell-mediated immunity. And third is the impact on the stress or anti-stress property. Now, coming to the adaptogen property, the name itself means that something which will help these animals to adapt within their environment. So what Phytosee is doing, and when you saw the whole physiology of stress, the most important link with all the form of stressors is that the body tends to increase corticosterone inside it to fight with the stress. While the amount of corticosterone keeps on increasing, it does not do any good to the body, rather starts harming the body. Similarly, what Phytosee will do as an adaptogenic property, Phytosee will bring down this corticosterone back to the normal, even if the stress is there. And then it will maintain the level of this corticosterone, even if the stressors are there. This is the adaptogenic property of Phytosee. You can see here what we try to do. We try to induce heat stress in the animals and measure the serum corticosterone level. And when Phytosee was given, the corticosterone level came back almost to the normal, but more importantly, the corticosterone level stayed and maintained at the same level even when the stress was there. This is the adaptogenic property of Phytosee, and this adaptogenic property is only possible because of the phytocompounds which are present in Phytosee. The phytocompounds which are present in Phytosee synergize with each other and have multiple target effects inside the body, thereby producing an impact which is called as an adaptogenic property. And what will happen? When the animals are adapted and the corticosterone level is controlled inside the body, the impact of multiple stressors will be minimized. So you can correlate that with reduction in the aggressiveness in your herd, reduction in the the impact which is created by the heat stress because of the social stress, or reduction in reproductive failures, which can be possible because of the multiple stressors. Now, going beyond, what we try to do, we try to test the adaptogenic property of Phytosee in our lab animals. So we did one test which is called as a swim endurance test, where we induced stress on the albino rats, and we try to see how much they can resist that stress when they are given Phytosee. If you see here, there are three groups. One is the stressed group, the orange one is the Phytosee group, and the blue one is the normal group. When the stress was given, we wanted to see how many seconds out of 300 these rats can swim in the water. The stressed rats could swim only for maximum of 239 seconds out of 300, while the same rats which were supplemented with Phytosee but were stressed could swim for much more time and almost equivalent to the control group, which means that Phytosee was able to increase the strength and resistance of these lab animals, increase the endurance of these lab animals towards the stress which they were facing. Similarly, what we did one more test in the zebrafish. We wanted to see how much endurance the zebrafish can have for a specific type of toxicity which we induced. If you see in this experiment, we cultured these zebrafishes for around 14 days in two different groups. One is the control group, another one is the treatment group. And after 14 days, on the 15th day, we induced ammonia toxicity in them. And then we checked how many fishes will survive and how much time they will take, for how much duration they will survive after the toxicity is induced. If you see the blue graph, the fish, the zebrafish which were not supplemented with Phytosee, and after inducing the toxicity, within 3 hours, all of them died. While as the group which was supplemented with Phytosee, you can see around 14 hours they took to resist that toxicity which was induced because of the ammonia on the 15th day. Now, this zebrafish model has become one of the standard models, an ideal model to study the stress responses and correlating that with the animals. The reason being, there is a huge genetic similarity for the zebrafish with the human beings or with the animals for that matter. They have the functional homology with the mammals and their whole genome sequence is present. Very importantly, the neuroendocrine responses in zebrafish are very robust and can be correlated strongly with the behavioral endpoints. So this is one of the reasons why, or possibly one of the reasons why zebrafish model is now becoming one of the important, or ideal models to study the stress response and correlate that with the animals. So by this study, what we wanted to see was that Phytosee has a very strong and potent adaptogenic property. Thereby, it increases the resistance and endurance of the animals. Now, coming to the second property of Phytosee, which is its impact on the cell-mediated immunity. Phytosee has a direct impact on the natural defenses of the animals. So it helps in the better proliferation of lymphocytes. It helps in the more proliferation of lysozyme, macrophages, and natural killer cells. And all of these are the first line of defense the body will, initiate against any form of stress. So what Phytosee is doing, Phytosee will increase the natural defenses, the first line of defense, and thereby, when the natural defense is, optimized, the acute defense would automatically be, improved upon after supplementation of this product, Phytosee. So what we did as a further validation to our, impact of Phytosee on the cell-mediated immunity, we did some more, validations like a neopterin test. For example, neopterin is a very important biomarker to see the impact of, to see the immune status of the animal. More is the neopterin in the neopterin level means better is the immune competence and immune status of the animal. So if you can see here again, the normal control, when we check the neopterin level, it was around 7.23 nanomole per liter. When the stress was induced, this neopterin level was reduced because the immunocompromised state, which was produced because of the stress, reduced the neopterin level. And the same group, when supplemented with Phytosee, the neopterin level was again significantly increased, which means that Phytosee was increasing the natural immunity of the animal, increasing the immunocompetence of the animal. Similarly, we did one more test, which is called as Cutaneous Basophil hypersensitivity test, which is same like tuberculin testing, where we have injected certain phytoantigen under the skin of the animal and checked what is the percentage response towards that antigen. More will be the percentage response towards the antigen, better will be the immune status of the animal. So if you see again, when the stress was given, the response was reduced. And when Phytosee was supplemented, the percentage response increased subsequently. Last but not the least, the last mechanism of action for Phytosee is that it acts as an internal cellular antioxidant. It increases the level of, natural antioxidants which are present inside the cells, like superoxide dismutase, catalase, and thereby rejuvenates the cell, reduces the free radicals inside the cells, makes the cell become more rejuvenated, and helps in the better absorption of the nutrients from these cells, which had become fatigued or old because of the increased metabolic rate of these cells. So coming to the overall features of this product, so what Phytosee will do in pigs, Phytosee will help in building up a strong and robust resistance or endurance of these animals towards the stress, and not only the heat stress, multiple form of stressors which these animals are encountering. Phytosee will help in building up a strong natural immunity of these animals by mobilizing the resources which are required for ensuring the natural responses are profound towards any stress. Phytosee will also help in the adaptogenic property by helping the animals to tolerate that stress. Even if the stress is not removed, the animal will try to tolerate and adapt to that stress. Last but not the least, one of the properties of Phytosee is that it is completely stable and can withstand the temperatures of pelletization of around 85 to 90 degrees Celsius. So it is completely thermostable. Now, coming to the trials, what we have, lately from last 2.5 years, we have been aggressively trying to, see the impact of Phytosee in swine. And we have done a lot of trials in the European markets, in different farms in the Europe, more specifically in Spain and Portugal, where we have seen a good amount of response towards this product by, working out on certain parameters of stress. The first trial, what I am going to showcase here is the trial which was done in Spain. Now, this trial was done in the pregnant sows, and the reason why this trial was done in Spain, after the sows are inseminated, they are kept in the separate, stalls for around 35 to 40 days till the proper implantation of the fetus takes place in the uterus. After 35 to 40 days, these sows are mixed together with the other pregnant sows. And because of the mixing of these sows, and because, as you remember, the social stress is one of the prominent factors which comes into play, there was increase in the aggressiveness of these animals. And because of the increase in the aggressiveness, they were fighting with each other, they would put bruises on each other, and it also had an impact on the reproductive functionality of these animals. So we wanted to see if Phytosee could reduce that stress and make these sows more calm. So what we did, we tried to check the marker of stress in these animals through the measurement of cortisol in the saliva. If you see this paper, the amount of cortisol in the saliva is a very prominent measurement of stress in these animals. Between 2 to 6 nanogram per ml of cortisol in the saliva is considered to be, okay for the animals. But as the amount increases, it goes beyond 6, from 12 to 40, it means that the animals are severely stressed. So same kind of thing we tried to extrapolate from this trial. We divided the animals into 2 groups, and we checked, we supplemented Phytosee for 5 days to the sows before mixing them together, and we checked the salivary cortisol level using the ELISA. Now, this is the whole design of this experiment. For the 5 days, these pregnant sows were kept in the separate pens. And after 5 days of supplementation of Phytosee, they were mixed together. And during the time of mixing, we wanted to see how much reduction in the stress or aggressive behavior in the sows would be possible. And we checked the cortisol level in the saliva. So if you see 5 days before mixing these sows together, in the control group, the average cortisol was around 3, and in the Phytosee group, the average cortisol was around 3.5 nanogram per ml. After 5 days, just on the day before they were to be mixed together with the other sows, the salivary cortisol level was again measured. Because the Phytosee was supplemented for 5 days, the Phytosee supplemented group showed a significant reduction in the salivary cortisol level in comparison to the control. Now, these sows have been mixed together, and there is an obvious response what will be there, that the cortisol level will be increased. So we checked 24 hours after mixing, what is the impact on the cortisol level in both the groups. And if you see here, the control group, the cortisol level was significantly higher, on an average around 33 nanogram per ml, while in case of Phytosee supplemented group, it was almost 50% less than the control group. And the animals were more calm. There was less aggression which was shown by the animals which was supplemented with Phytosee. So the whole study showed that Phytosee was able to control the levels of cortisol inside the body of sows at the time of mixing, thereby reducing the aggression in these animals. Now, the second trial which we did was done in Portugal, and we wanted to see in this trial what would be the impact of Phytosee on the post-weaning stress. as post-weaning stress is one of the most critical times for the pigs as they move from the mother's milk to the solid feed, and they are removed from the sow. It is one of the critical times for the piglets. And during this period, the mortality levels also increase, and the aggression, like tail biting and ear biting, are also very high, because you're mixing these piglets together in the nursery, and unfamiliar piglets are mixed together, which invokes this stress of aggression in them. So in this experiment, what we did this experiment almost 10 days after weaning the piglets, and we divided these piglets into two houses, H1 and H2 house, and almost we did this experiment for the whole of the until the growing period started. If you see the impact of Phytosee on the body weight, at the time of weaning, both the houses, the piglets were almost of the same weaning weight. But at the end of the experiment, after 58 days, the groups which were supplemented with Phytosee in both the houses, they showed good impact on the overall average body weight of the herd. Number two, the average daily gain in both the houses supplemented with Phytosee was much better than the groups which were not supplemented with Phytosee. Last but not the least, the mortality percentage in both the houses supplemented with Phytosee was reduced when, in comparison to the groups which were not supplemented with Phytosee. In addition to that, the vices of ear biting or tail biting were significantly reduced after the supplementation of Phytosee. Coming to the third trial which we did, and this was one of the latest trials which we have done on the effect of Phytosee on the sows during the later part of the gestation and the lactation. So what we wanted to do or find out in this experiment was how much Phytosee will be able to reduce the stress at the time of furrowing in the sows, and how much Phytosee can improve the quality of the colostrum and the milk by increasing the immunoglobulin levels coming from the sows. So if you see the design of this experiment, we divided the animals into two groups, control and treatment. And we took the salivary cortisol estimation was done 7 days before parturition in the sows, then on 2 days before parturition, on the day of parturition, and 7 days after parturition. Because 7th day after parturition is the time when the immunity, passive immunity, reaches to its peak in the piglets. Similarly, in case of piglets, which were lactating the colostrum and the milk from the sows, we checked the blood as well as the colostrum for the immunoglobulin content, which whether it's increased or decreased after supplementation of Phytosee. So if you see how Phytosee was able to modulate the cortisol levels in the sows 7 days before parturition, the control and treatment group, there was a significant reduction in the Phytosee supplemented group. Even on the day of parturition, on the day of furrowing, which is the peak of stress for these sows, you can see there was a significant reduction in the cortisol level, thereby making sure that these sows pass through this furrowing period without much stress, which also means that the amount of immunity which can be transferred through the colostrum and through the milk to the piglets will be increased subsequently. And which is what actually happened. We checked the immunoglobulin G levels in the blood of the piglets on the day 7 and the colostrum of the sows. If you see again, there was a significant impact on the IgG levels, both in the blood of the piglets or in the serum of the piglets and in the colostrum of the sows, thereby showcasing that Phytosee increased the immunocompetence of the pregnant sows to activate the immune system to produce more antibodies and produce more immunoglobulins, which can be transferred from the milk to the piglets. So Phytosee was able to increase the quality of the colostrum, as well as it was able to significantly increase the immunoglobulin levels in the piglets, and thereby making these piglets more immunocompetent. Now you can imagine on the 7th day, around 54% increase in the immunoglobulin levels in the piglets. It will mean that the same herd of piglets will reach to the weaning in the better possible way. There'll be maximum number of piglets which will reach the weaning than the piglets which were not, which were not having enough of immunity to withstand the stress during this period. In conclusion, Phytosee, there are other reports what we have, the impact of Phytosee on controlling the panting in sows, but these are more of the feedbacks, and we are just trying to collate the whole data. Phytosee, if you see the general observations, Phytosee is able to control the panting. Phytosee is able to have improve upon the better health and weight in the finishing stages of the pigs. Also, it will have a good impact on the meat quality of the pigs in the finishing stages. It will reduce the parturition stress, will impact the the immunity of the piglets, thereby ensuring more number of piglets are there at the time of weaning, and it will help in the reduction of the post-weaning stress as well as the pre-weaning and post-weaning mortality. So coming to the last part, what are the benefits of Phytosee? In case of piglets, as I just mentioned, Phytosee will improve on the piglet immunity, mostly the passive immunity coming from the sows during the lactation period. And also, in case of piglets, it will help in reducing the weaning stress when the piglets are weaned at around 21 days. So it will also mean that there is reduced mortality at the time of, for the first week of weaning and after weaning, and there will be more uniform body weight and the uniform body condition of these piglets. In case of sows, Phytosee will help in reducing the stress, which is very prominent at the later stages of the gestation, just 10 to 15 days prior to gestation, prior to furrowing, and it will help in improving the quality of the colostrum and milk with more, immune cells coming into the milk, thereby improving the immunity of the piglets. It will also help in reducing the social stress which will be created at the time of regrouping of these sows after weaning and after insemination of these sows. It will reduce also the impact which heat stress can have, and thereby Phytosee is able to produce better quality litter, better size of litter, and better weaning weights, and improves upon the fertility rate of the sows. In case of fattening pigs, as I just mentioned, we are in the process of doing certain trials, and we have seen that Phytosee is able to reduce the stress which is created by the transportation and at the time of slaughter. Thereby, it helps in the improvement of the quality of the meat by restoring the amount of glycogen which is necessary to maintain the pH of the muscles. In case of breeding boars and gilts, when Phytosee is supplemented, it will help in the better quality of the semen and even the better quantity of the semen, and it will increase the libido and fertility of both males as well as females, in addition to fighting the stressors which will be there at the time of breeding of these boars and gilts. Coming to the dosage and of this product, what we have done, we have divided the dosage of this product right from different phases of the rearing cycle of the pigs. So the dosage would range from around 750 ppm to 3000 ppm. So in different stages, like in the post-weaning stage, the dosage is around 1.5 to 2 kg for around 20 days. Similarly, in case of sows, during the gestation, in the early gestation, before mixing, the dosage can be around 1.5 to 2 kg. And just prior to the furrowing, around 10 days before furrowing, the dosage can be increased up to 3 kg to improve the immunity of the piglets subsequently. During lactation, since the feed intake is very high, the dosage will vary between 750 to 1000 gram per metric ton. And in the fattening and finisher pigs, 1 month prior to the transportation or 1 month prior to the slaughter, we have seen that a dosage of around 1 to 1.5 kg will have a positive impact on the overall quality of the meat. Lastly, in case of breeding boars and gilts, the dosage of Phytosee will be around 2 kg, and it will have an impact on the overall quality and the quantity of semen, in addition to improving upon the fertility of the animals. So that was all about this presentation. If you have any questions, you can write down in the chat box and we'll take up the questions. Furthermore, if you have any further questions, you can write down those questions and share it with us on our respective email IDs, which we will, which you are seeing on the screen right now. Thank you very much. I'll now stop sharing my screen and we'll take up some questions from the audience. Thanks a lot.
Thank you, Junaid, for a very clear and straight presentation. I must say that you cover very efficiently these topics because you just stood in the hour. The hour is gone right now, but we do have some questions, and both in chat and in question and answer session. So I think you can see them. Some of them are very, very quick, like very public, like the one posted by Michele. They run a trial already similar to the one you just presented, I guess, with Phytosee, indeed. Mm-hmm. And they have said a huge variation in responses in colostrum. Do you think that parity can play some role in this homogeneity?
Of course, I think more is the. So you mean to say what would be the impact of parity on the quality of colostrum?
Yeah, the response to this kind of stimulation.
Absolutely. I mean, the more these animals move towards the parity, I mean, the larger is the parity, the immune functionality of the animals would be more, compromised because the animals are moving towards, older age. So what we have seen that Phytosee supplementation in these animals in the later part of their, in the larger parity stages of these animals has a good impact on the, on activating certain immune, functions inside the body of the sows. Like we have seen that Phytosee has a good impact on the activation of, gut-associated lymphocytes, and Phytosee has a good impact on the activation of certain, lymphocytes which are present in the respiratory epithelium, which in fact helps in the better transfer of immunoglobulins into the colostrum. So to say or to answer this question, yes, in the old parity sows, when Phytosee will be supplemented, it will definitely have an impact on the quality of the colostrum, which is, dwindling with the age of the sows. So when you supplement Phytosee to these sows in the larger parity, the quality of the colostrum and the transfer of immunoglobulins and the transfer of maternally derived antibodies will be more optimized than the sows of the same parity without the supplementation of Phytosee.
Okay. Thank you. Very general question, a very practical, asked by quite a few people. Can you clarify the dosage you used in the trials that you showed in all of them, sows and pregnant, okay, lactation, and piglets?
All right. So what we did in these trials, in these commercial trials, we did not add Phytosee directly in the feed. We added Phytosee as a top-up because we wanted to see what would be the impact of adding Phytosee as a top-up. So what we did, we added a dose of around 20 gram per sow per day in most of the experiments. The reason going to this higher dosage was because we were manually going to add this Phytosee for each of the sow, and there would be a possibility of error in the dosage, actual dosage being, given to the sows, and also a part of the, feed also gets sticked to the snout of the piglets or the pigs or sows. Thereby, we gave the dose of around 20 gram per sow per day. But having said that, once we found the results, we found out that the dosage which we have given at the end of the presentation, let's say in case of lactating sows, we are seeing that a dose of around 2 to 3 kg is giving you the equivalent response, which we found out by giving the dose of around 20 gram per sow per day. Similarly, In case of sows, just at the time of mixing them, 40 days after insemination, a dose of around 2 kg per ton of feed is giving you the equivalent results as was given by 20 gram per sow per day in the experiment. So the dosage of 20 gram per sow per day was only because we were using it as a top-up, and we understood that there would be certain error in the actual, collation of the dose, and there will be some loss of the feed which was mixed with this Phytosee. So yeah, so dose of around 2 to 3 kg in the later gestation prior to furrowing, and 2 kg in the early gestation just before the mixing is what we are commercially using now in many countries.
Thank you, Junaid. I think we are running out of time, but I guess a couple of questions, quick questions, I can still share with you. One is, can stress affect back fat quality? And if yes, how?
Actually, yes, because stress will have a lot of impact on the back fat quality as well. As if you go through the presentation which I presented, whenever the stress is increased inside the body, what body tries to do? Body tries to increase the emergency sources which are there inside the body, to increase the amount of energy in it, to increase the amount of glucose in it. Now, the most easiest source of energy for the body, or the immediate source of energy for the body, is the glycogen which is present. But if the stress keeps on increasing, the glycogen reserves are completely depleted, and body will go towards the lipids finally to break them down and transform those lipids into glucose as a source of energy. So yes, stress will have an impact on the on the overall fat reduction in animals, but it's not a positive sign actually, because this is the fat which is these are the fat reservoirs which are important for the body, and the body is itself using these fat reservoirs as a source of energy to overcome the stress which it is facing. Coming to Phytosee, whether it will have an impact on this, we do not have any validation so far whether Phytosee has an impact, but yes, as a stress, it definitely has an impact on this parameter, and it reduces a lot of fat, because the chronic stress will create these kind of problems.
Thank you. I think this is the very last question. Can Phytosee affect immune response to vaccination?
Actually, this is a very good question. See, Phytosee basically tends to improvise the natural defenses of the animal. It is very obvious to correlate. If the natural defense of the animal would be optimized or upregulated, what will happen? The body will be in less stress. The body will be in optimal condition, which will therefore translate into better acute immunity. Okay, so what will happen if you're giving Phytosee, your natural defenses are optimized? There is a very strong possibility that your vaccination, antibodies which are produced because of vaccination would be improved after supplementing Phytosee. Although we have not done the trials in swine for such parameters so far, but because there is a strong possibility and there is a strong correlation for this, as the natural defense, as the resistance of the body increases, there is a strong correlation that the acute defenses of the body will also be increased. So yes, there is a good correlation. Phytosee can improve the titer levels inside the body, but again, this would be completely depending on the dose regimen which we will follow for this product. There is a specified dose regimen and the phase in which we want to use, and for the time this product is used, will finally decide the amount of impact it will have on the acute defenses, or the impact after the vaccination.
Thank you. This is, I just say that it was the last one, but I see a new one which is very strictly related to this last point. So what is the time that it takes for Phytosee to start showing effects on a standard use in feed?
Correct. So with our all experiments in swine, we have seen that Phytosee will start showing its tangible benefits within 5 to 7 days of its supplementation. So within 5 to 7 days, you will start finding the very quantifying benefits of Phytosee. For example, the trial which we did in the gestational sows, which were mixed after 40 days of insemination, for 5 days only, we supplemented Phytosee, and when we grouped these animals together, we could see that there was a significant reduction in the aggressive behavior of these sows, which means that Phytosee was able to showcase its effect within 5 days. So we say that within 5 to 7 days, the product will start showing its visible, effects and benefits, which will be, easy to quantify by anyone.
Okay, thank you. Very clear. And I think this was really the last question because we are running out of time. It's time is gone, but it was a very clear and effective presentation. So Junaid, let me thank you very much for your very helpful support. And I hope that we might find in the future new opportunities for new webinar, maybe to find new topics, agree with our customers to explore. And for the moment, I would also like to thank all of our guests, our customers and people that join us today for the very active participation, questions, and of course, again, our contacts were shown. Because you got a mail with invitation, so the contacts are still there. For any further questions, please let us know. And thank you again for participating. And